Welcome everyone to this month's cross-disciplinary seminar series on metabolic disease. Today I have the pleasure to introduce Dr. Lev Becker. Dr. Becker received his Ph.D. from Queen's University before conducting his postdoc at the University of Washington. And he then joined the faculty at University of Chicago in 2012, where he's currently serving as an associate professor in the Ben May Department for Cancer Research. Dr. Becker's research focuses largely on myeloid cell dysfunction that occurs in chronic sterile inflammatory states such as obesity, diabetes, cancer. In particular, a lot of his work focuses on covering functional changes in neutrophil and macrophage populations that contribute to the underlying pathologies. So his lab is highly productive. Ever since he started, he's really been quite well published in high impact journals. His research has also been quite well funded, receiving multiple R01s in addition to other various grants. I have to say, in addition to being an excellent scientist, Lev is known in his circles for being an incredibly collegial, kind, and generous person, which I find very admirable in sometimes the cutthroat world of biomedical research and academics. So Lev, I'm very excited for you to present in our cross-disciplinary seminar series today. So welcome to GW.
Thank you so much, Brett, and thanks again for the invite and also for the very kind introduction. I'm going to share my screen here. Please let me know if you guys are able to see that.
It's good.
Okay, great, and I'm really looking forward to a great discussion following the seminar as well. So today I'm going to tell you a little bit about the work that we're working on in the lab focused on attenuating the deleterious inflammation during metabolic disease. And I'm going to start off by saying that, I think it's generally thought that inflammation is a bad thing, and it certainly is in the chronic state, but we also need to appreciate that we absolutely require acute inflammation to help combat various infections, bacterial or viral ones. And so an appropriate response is having this acute inflammation go up and then dissipate. However, in the setting of chronic inflammation, there's a multitude of diseases that can result from that. And that's kind of shown here in this slide. All of the sort of chronic inflammatory states and diseases that have been associated with it, as well as the drivers of that chronic inflammation on the left. And so in the talk today, we're going to touch on obesity, infection, as well as metabolic syndrome and cancer, and importantly, start trying to create mechanistic links, as well as showing mechanistic disparities between the inflammation that takes place in these very diverse contexts. And so taking a step back, historically, macrophages, which are one of the primary cells that are responsible for initiating and attenuating the inflammatory response, has been thought about from this old paradigm of these M1 type macrophages or these M2 type macrophages. The M1 macrophages are triggered by things such as bacterial products, interferon gamma, and they support this immunogenic, pro-inflammatory, and sometimes tissue destructive state, while the M2 type macrophages are driven by anti-inflammatory cytokines such as IL-4, IL-10, and they are responsible for mediating tolerogenic, anti-inflammatory, and tissue regenerative type pathways. Now, it's not that one of these cells is good or bad. It's really about context. You really need the M1 to fight infection, and you need the M2 to support wound repair, but it's the M2s that are pathological in cancer, while the M1s are anti-cancer. Whereas in obesity, it's the exact opposite. It's more of these pro-inflammatory cells that are problematic, while the anti-inflammatory ones are helpful. Now, this understanding in particular of the inflammatory macrophage in the context of bacterial exposure has really been used to understand inflammation in sterile inflammatory diseases, such as obesity, cancer, arthritis, amongst others. And it's very much true that the cytokines that you observe to be induced by exposure to bacteria, such as TNF, IL-1, IL-6, are also induced in these chronic conditions, albeit at a much lower grade and a much longer tenure. And one of the challenges of approaching the concept of inflammation from a bacterial model is that, in my view, it really limits the development of therapies to target this inflammation. And the reason is if you go after these cytokines, which is what many have attempted, that these cytokine inactivating antibodies will not differentiate between how you got inflamed. And so, for example, an anti-TNF that is used to treat obesity is also going to antagonize TNF when we are exposed to pathogens. And from a therapeutics development perspective, this is problematic because patients can be predisposed to opportunistic infections. And also, we can only target perhaps one cytokine rather than all of these cytokines due to that risk of these opportunistic infections. And I want to push out the idea that if we start thinking about inflammation in a different way and let go of some of the knowledge that we've obtained from infectious inflammation, perhaps we can get to a more specific and safe and more efficacious way to inhibit inflammation. And so in this context, if you identify mechanisms that are driving inflammation in obesity, and those are different from an infectious state, one can then target that pathway to attenuate all of these cytokines and do it in a specific way that does not in any way predispose to infection. And so the story I'm going to share with you today is more about the history of how we got here. We're actively developing these things, but it's still a little premature to share the data that we have on this topic. And so, when we started this project back in 2011, this was kind of the model of what we knew about macrophage phenotypes during obesity. The idea was, in a lean state, you had these anti-inflammatory M2 type macs. And then as the adipose tissue expands during obesity, you get two things that happen. One is that there's an infiltration of macrophages into fat. And then the second is this sort of chronic M1-like pro-inflammatory cell phenotype that's acquired by those adipose tissue macs. And so at that time, we had discovered a new cell phenotype, which we coined metabolically activated macrophages, or MME for short. And I'll just share a little bit of the data that was some of the early data that really perhaps suggested that while the macrophages in adipose tissue are inflamed, they don't resemble these bacterially challenged M1 type macrophages. And so at the time, there were very poor cell surface markers to identify different phenotypes of macrophages, in particular in humans. And so what we did is we took human monocyte-derived macrophages, and we either made them with MCSF, these are designated by M as the first letter, or by GM-CSF. These are two different ways that macrophages can differentiate in vivo. And then we polarize those macs to either the M1 or M2 state. And the idea here was to define a set of cell surface markers that reliably identified M1 type macrophages independent of how that macrophage was differentiated in which cytokine milieu. And so we kind of identified this pattern of cell surface receptors, and this was all done by plasma membrane proteomics. So we wanted to validate that these plasma membrane proteomics could be extended to flow cytometry, which is of course how we'd be studying these in vivo in patients. And indeed, we validated all of these markers to be specific to M1 type macrophages in vitro polarized human macrophages. So armed with these markers, we went into obese patients. We looked both in subcutaneous and omental fat depots. And we could see that, these depots were indeed inflamed, as has been widely reported by many groups. TNF alpha and IL-1 beta levels were elevated. However, we did not observe any positivity for those M1 markers. I'm showing you one of them, but we tested many of them. And the cells, the pro-inflammatory macrophages from patients in their visceral or subcutaneous fat were just not expressing any of the proteins diagnostic of M1. The same was true in obese mice. Again, macrophages are inflamed, but they're not expressing anything that the M1 type macrophages should be expressing. So that observation could be explained one of two ways. The simplest explanation is that our markers just aren't very good. They were generated based on in vitro systems, and in vivo, things are more complicated. The other interpretation is that the adipose tissue MAC phenotype just doesn't resemble a macrophage that saw a bacteria. And so to differentiate between these two ideas, we decided to get macrophages out of patients with cystic fibrosis. And these patients have chronic bacterial infections, and you can see the microbiology of the sputum indeed demonstrated that. And when we looked at patients that had chronic bacterial infections, these markers all lit up very nicely on the macrophages. And so this data kind of told us it's not that these markers are bad per se, it's that they're more appropriate for macrophages that are being exposed to infectious type stimuli, not metabolic stimuli or sterile inflammation. And that's kind of shown a little bit more closely here across these three markers. You can see that these proteins were very low expression and not elevated in patients with obesity, while they were very clearly and highly detected in the patients that were exposed to bacteria in the cystic fibrosis airway. So that kind of led us to conclude that perhaps metabolic dysfunction was not supporting this M1 type phenotype. And we started asking, well, what might be driving the inflammatory state of macrophages during obesity? And we decided, after looking at the literature, that molecules such as glucose, saturated fatty acids, and insulin all had been reported to induce inflammation in macrophages, at least in vitro studies. So we combined this cocktail and put it on macrophages, and we refer to cells exposed to this as metabolically activated, or MME. And so when we first started, we wanted to compare how different does an LPS interferon gamma challenged macrophage look in comparison to metabolically activated macrophage. And what we observed was actually very consistent with what you see in vivo. So cytokines were elevated by both perturbations, but you can see that the extent to which cytokines are induced is substantially lower in the metabolically activated macrophages than the classically activated M1s. And this is, of course, what's observed in vivo. During infection, you get very high increases in cytokines, while obesity is characterized by a chronic low-grade inflammation. We further looked at the cell surface markers that we had identified and observed similar results to what we saw in the patients, which was that while the MME macrophages were inflamed, they were not expressing those same markers of M1 in this in vitro system. So because they didn't express those markers, we then wanted to ask, what markers do identify these MME type macrophages? And more broadly, how different are these cells? And so we did a proteomics analysis on the plasma membranes of MME and M1 macrophages. And I always find this plot to be really illustrative of how different these cells are. What you can see is that the proteins that are either down-regulated in M1 or up-regulated in M1 are pretty much orthogonal to those that are up or down-regulated in MME, meaning that these cells, while they share induction of inflammation, almost everything else is different between them. And when we looked more closely at what proteins were elevated specifically in the MME type macrophages, the theme of induced lipid metabolism came out very clearly from the proteomics data. And this, of course, makes sense. The macrophages are sitting in adipose tissue in a lipid-rich milieu, so it makes sense for them to induce proteins that would help them to either internalize or metabolize or efflux lipids from the cells. And right around the same time that we published this paper, work from Tony Ferrante's lab at Columbia published something very similar that in mice that adipose tissue macrophages were characterized by increased lipid metabolism independent of classical activation. And subsequent to our two papers on this topic, there's been a host of papers that have come out since that time that have also identified this induction of lipid metabolism and these lipid-loaded macrophages in obese patients as well as obese mice. So we now had some new markers based on ex vivo experiments, and we went back to our patients and asked, do these markers now identify these pro-inflammatory macrophages in the patients? And again, you can see we looked at omental fat and sub-Q fat, and we could see the induction of these proteins on the cell surface of the adipose tissue macrophages. And just to demonstrate specificity, we also looked in those CF patients, and those same lipid metabolism proteins were either not induced or even suppressed. And then finally, we could correlate the cell surface levels of these proteins in adipose tissue max with the extent of adiposity, while the M1 type markers, which were very lowly expressed or not expressed, also did not correlate in any meaningful way. And so those are correlations that were done with BMI, and we had no clue what the insulin resistance status of our patient cohort was in those studies. So more recent work, and this is a paper we're about to submit, really asked the question whether these MME-like ATMs correlate with insulin resistance in patients. And so to do this, we recruited patients that were matched for adiposity, but differing in insulin sensitivity. And you can see that the patient cohort here were deemed insulin resistant based on HOMA-IR. That was the definition. But we also observed significant decreases in the Matsuda index, as well as in OGTT studies, where we looked at glucose tolerance as well as insulin levels during the glucose tolerance test. And we could show that the patient, the insulin-resistant patients, did have elevated glucose, or sorry, elevated insulin to maintain that same glucose level. And so the interpretation here is that the subjects are really insulin-resistant but pre-diabetic. But most importantly, you can see that the BMI, percent body fat, and waist to hip ratio of these patients was very well matched. So we could now ask what correlations these MME-like adipose tissue macrophages have with insulin sensitivity independent of changes in adiposity. And those parameters are shown here. We looked both in the omental adipose tissue depot as well as the subcutaneous adipose tissue depot, and thematically they basically showed the same thing. These MME-like adipose tissue macrophages were positively correlated with both HOMA-IR as well as the insulin area under curve in the OGTT study in both adipose tissue depots. And this was interesting that although we observed correlations in the phenotypes, we did not observe correlations between adipose tissue macrophage number or these crown-like structures, which are macrophages that surround dead adipocytes. And I'll show you more data on that in the subsequent slides. But these are two parameters that have been previously reported to correlate. We did not observe them in our patient cohort, while we did observe correlations with the MME. And so all of those studies established the presence of MME-like macrophages in adipose tissue, but we're also interested in their function. And so we started to look at what role these cells might be playing during obesity-associated insulin resistance. And this is kind of a summary of the mechanistic insights gained from our 2014 paper. And what we showed here was that the free fatty acids in the MME are able to signal through cell surface receptors to drive NF-kappa B activation, but those fatty acids also get internalized to induce signaling through P62 or PPAR gamma. And it was this signaling cascade that induced the lipid metabolism and actually lowered NF-kappa B activation. And you can see this mechanism is quite distinct from what happens when cells are treated with bacterial products such as LPS. And so in order for us to gain insights into what MME macrophages were doing, we needed genetic perturbations that selectively inhibited inflammation in MME type macrophages, but not in M1 type macrophages. So we got several knockout mice based on the literature, and we tested these knockout mice for their ability to inhibit inflammation in these two different cell types. What we found was that while TLR4 and MYD88 inhibited inflammation in both cell types, TLR2 and NOX2, or NADPH oxidase 2, only inhibited inflammation in the MME, while IL-1R and IL-18R had no impact on either. And so this is just data showing you for the NADPH oxidase knockout. We knocked out the GP91 subunit in order to eliminate the enzyme. And you can see in reductions in cytokines in the MME state with no impact in the M1 state over on the right. And consistent with this idea, previous work from Ira Tabas's group showed that oxidized lipoproteins also activate macrophages through NADPH oxidase 2 and TLR2 based mechanism. So these proteins may be involved more broadly in lipid stress of the macrophage rather than infectious type stimuli. And so in unpublished work, we are also able to show that if you just look at the co-localization of NADPH oxidase 2 and TLR2 in the M0 ground state or in the M1 activated state, we do not observe changes in their cell distribution. However, only 60 minutes after adding MME stimuli, you can see that the proteins now start co-localizing and perhaps are signaling in order to activate the cells. And remarkably, when we did a cytokine array on macrophages that were either TLR2 knockout or NADPH oxidase 2 knockout, these two perturbations had very strongly overlapping effects on cytokine release into the media by MME macrophages, indicating that they are functioning perhaps as a collective, and which is consistent with the binding data here, or colocalization data on the left. So eliminating one blocks signaling, eliminating the other blocks signaling in the exact same way. And both of those perturbations, as I said, were specific to MME type macrophages. And so with that in mind, we had these myeloid cell GP91 knockout mice, and we put them on high-fat diet. And the first thing that we observed was that the animals that were knockouts tended to be heavier. And so the data I'm going to show you here is at the 8-week time point, and you can see either on the high-fat diet or the low-fat diet, the GP91 knockout mice gain more weight. However, when we looked at inflammation in adipose tissue macrophages that were purified, we could see, just like we saw in vitro, that the GP91 knockout was able to attenuate inflammatory cytokine expression in the adipose tissue macrophages. And we also observed that this amounted to improved glucose tolerance in the mice. We did not see an improvement in insulin resistance. And so at the 8-week time point, so we decided to look at the 16-week time point. We thought maybe we'd get more pronounced differences at that duration of feeding. And So at the 16-week time point, just again mentioned that the mice or knockout mice are more obese in both low fat and high fat. However, much to our surprise, the glucose tolerance now was actually no longer improved, it was equivalent. And when we measured insulin in these mice, we noted that the knockout mice needed more insulin in order to maintain this same glucose tolerance, suggesting insulin resistance. And we could test that more directly through the insulin tolerance test, where again, you can see that the knockout mice actually develop quite severe insulin resistance after 16 weeks of high fat feeding. So we had this knockout that seemed to have beneficial effects early during the diet and then have worsening effects later. And we were interested in trying to understand how that was taking place. And so the first thing we wanted to rule out was the idea that the worsening of insulin resistance was just due to increased body weight. So we basically took the wild type mice and we let them go on diets a little bit longer to weight match them. However, weight matching did not increase the insulin level. So we could rule out the idea that adiposity was causing this. Next, we took a look at the livers of the mice. And what we observed was that the knockout mice on the high-fat diet at 16 weeks had severe lipid accumulation in the liver. And if you take a look at just liver weight over here, the livers of some of these mice were twice the size of control mice. And this is not the normal hepatosteatosis that's observed during high-fat feeding, which is shown in the wild type picture right above. And so when we had measured gene expression in the liver, and there was no increase in lipid metabolism or lipid synthesis genes. And previous studies had indicated that one way you can get liver accumulation or liver fat accumulation is if adipose tissue becomes unhealthy. And so we started to look at visceral fat in the obese mice. And you can see in the wild type, these are those crown-like structures that I mentioned to you briefly before. These are many macrophages surrounding dead adipocytes. And you certainly see them in wild type mice fed a high-fat diet. But when we looked at the knockout mice, every single field had huge influx of these crown-like structures, suggesting that the adipose tissue was dying. And you can see that perilipin 2 is a marker of adipocyte death. And so when adipocytes die, their perilipin positivity goes away. And if you look carefully, you can see these crown-like structures are surrounding perilipin negative adipocytes, suggesting that they're dying. And we could confirm that by looking at caspase-3 activity in the adipose, which was greatly elevated in the knockout mice on the high-fat diet. And this sort of phenotype resembles lipodystrophy in some ways, where adipose tissue is no longer able to accumulate nutrients and store fat, and then that fat goes to the liver and insulin resistance ensues. However, this is not lipodystrophy. You can see the mice on 8 weeks are perfectly capable of expanding their adipose tissue. However, at 16 weeks, the adipose tissue becomes smaller in these mice, even though the mice are heavier. So clearly, there was no issue expanding fat. There was an issue in maintaining that expanded fat. And what we kind of started to realize was that the defect was happening because macrophages were unable to clear dead adipocytes. And so when we think about why dead adipocytes accumulate, we often think of positive triggers. And it's known that, for example, hypoxia and inflammation can induce adipocyte death in adipose tissue. However, when we looked at those parameters on the knockouts versus the wild type, we saw no increases in markers of hypoxia and no increases in inflammatory cytokines, suggesting that the accumulation of dead adipocytes wasn't being driven by increased triggers of death. And so we started to think about, is it possible that perhaps they're accumulating because the macrophages aren't able to clear the dead cells? And that raised the broader question of how does a macrophage clear a dead adipocyte to begin with? And that's an interesting question for the following reason. Macrophages are roughly 20 microns, and it's very easy to conceptualize how they can eat and engulf a bacteria or even bring in a dead neutrophil. However, clearing a dead adipocyte seems to be a tall task based on the size difference between these cells. And I kind of get reminded of this image that once went viral on the internet. I think it was a boa constrictor eating an alligator, or the other way around. I can't remember now. But you know, the meal that was taken in was too large, and the animal actually died trying to eat this larger creature, which of course is what would happen if a single macrophage tried to engulf a single dead adipocyte. And so at the time I met with Fred Maxfield from Weill Cornell, and he was working on this problem. And what he was showing was that instead of engulfing a dead adipocyte, the macrophage can actually fuse with the dead adipocyte and then release lysosomal acid lipase to liberate triglycerides into free fatty acids, which then get internalized by the macrophage and stored as lipid here. And of course, even through this external process, this is still too big a meal for one macrophage. So additional friends of that macrophage surround this dead adipocyte, and they're all releasing lysosomal acid lipase, taking the triglyceride and metabolizing it. And that's, of course, what we're observing here in vivo in these crown-like structures. You can see all of the different cells identified by their DAPI nuclei. Green is a macrophage marker surrounding this dead adipocyte. Fred had also developed a really amazing in vitro assay to study this. And in this assay, macrophages are loaded with biotin fluorescein dextran, which labels the lysosome. The adipocytes are streptavidin labeled and are induced to undergo apoptosis. And then when the apoptotic adipocytes are co-incubated with the macrophages, one can capture lysosomal release onto the cell surface of the adipocyte through that fluorescence dextran and quantify these events. After 1 hour, after 24 hours, you can also measure lipid accumulation in the macrophages that ensues using an oil red O stain. And so here's data now from the wild type versus the NADPH oxidase 2 knockout. And you can see that the knockout is able to deliver far less fluorescence to a dead adipocyte. And that's quantified in this graph here. And importantly, you can also see that performing this experiment with live adipocytes does not result in this same extent of lysosomal delivery. When we look 24 hours later, the inability of the GP91 knockout macrophages to exocytose their lysosomes was also visualized by the fact that they could not accumulate fat after being, sorry, I'm pointing at the wrong image, after being co-incubated with these dead adipocytes. So you can see in the wild type, all of these lipids that can be identified. However, in the knockout, those lipids are pretty much absent. And just as a control, we wanted to show that there was no defect in internalizing free fatty acids between these. And so the idea is they can't get the fatty acids because they can't liberate them from the dying adipocyte in the first place. Now, importantly, this process of lysosomal exocytosis is not actually directly related to inflammation. And we have two lines of evidence to suggest that. The first is that if we can't rescue the ability of the knockout macrophages to clear dead adipocytes by giving TNF alpha, one of the key cytokines made by them. And also, if we knock down inflammatory cytokine expression with TLR4 instead of NOX2, you can see we can lower cytokines, but the ability to exocytose the lysosomes is not impacted by that. And so this suggests that the signaling cascade driven by NOX2 will bifurcate, and one part of the pathway will be responsible for lysosomal exocytosis, and another part of the pathway will be responsible for cytokine expression. And so this is the summary of the work here, that in the early onset, we get this MME type macrophage, which produces cytokines leading to metabolic derailments, but that at late onset, those same MME type macrophages perform this key function of dead adipocyte clearance. And if that's blocked, you end up getting ectopic fat deposition in the liver, which then drives insulin resistance. And that may help to explain the divergent phenotypes observed after different times on the high-fat diet. Now, in addition to insulin resistance, we also wanted to investigate the idea that perhaps these MME type macrophages may serve as a mechanistic link between obesity and breast cancer. And this work was done in collaboration with Marcia Rossner here at University of Chicago. And the idea here is that, obesity is now one of the leading risk factors for the development of cancer, and is becoming a very large clinical problem in our society. And we decided to focus on triple negative breast cancer for several reasons. The first and foremost was that breast cancer happens in adipose tissue, and we know obesity directly alters adipose tissue biology. And the other reason was because this breast cancer is very aggressive and has very poor treatment opportunities. And so we thought, identifying new mechanisms here might also be more helpful from a translational perspective. There's earlier studies showing that in mammary fat, that adipose tissue macrophages do accumulate and form these crown-like structures in breast fat. And this is really important because different fat depots show very different biologies in obese mice and obese humans. So just because we showed MME were present in visceral fat doesn't mean they will also be present in mammary fat. Often there can be differences. But in this study, they not only showed that they accumulate, they could also correlate the crown-like structure density with metrics of adiposity, such as waist to hip ratio and BMI. And so our hypothesis was that, based on the idea that chronic inflammation, irrespective of its source, can induce a stem cell-like phenotype in cancer cells. And these stem cell-like cells have an increased ability to form tumors for self-renewal, as well as metastasis, although the latter has been subject to some debate. And so obviously viruses and bacteria are not driving chronic inflammation during obesity, but we thought that the chronic macrophage inflammation in mammary fat could be serving as a potential mechanism to feed into this cancer stem-like phenotype and drive breast cancer. And one sort of piece of background information that's going to be important is one of the key mechanisms by which cancer cells adopt this stem-like phenotype is through signaling through GP130, which can form heterotetramers with different cytokine receptors and signal through the JAK-STAT pathway to directly induce master regulators of the stem cell program. And so we started in patients that were either cancer-free, and this work was done with collaborators from Northwestern as well as from University of Chicago. And we isolated mammary fat from obese women that did not have cancer. And just like we observed in the visceral and subcutaneous depots I showed you previously, the MME-like phenotype was elevated in mammary fat. However, we did not observe very much M1 activation, and it certainly did not change between changes in BMI. And also, we could correlate the extent of these MME macrophages in mammary fat with BMI, but not M1. And then working with Funmi Olapati, we also got tissue sections from obese women with early triple-negative breast cancer tumors, and could show that if women were overweight or obese, they had evidence of more STAT3, phospho-STAT3 in their nucleus in the cancer cells in these early developing tumors. And so to kind of try to link this to the macrophages, we isolated the adipose tissue macrophages from the obese tumor-free women, collected their media, and then treated human triple-negative breast cancer cells in vitro, and could show, first thing, that in comparison to control, you can see the adipose tissue macrophage media induced STAT3 phosphorylation, and that induction could be blocked by eliminating that key cell surface receptor I mentioned earlier, GP130, on the cancer cells. We also wanted to test this in mice and looking at mammary fat. And again, mammary fat macrophages were more inflamed in the high-fat diet. And similar to what we observed in humans, we did not see induction of M1 type markers, but did see induction of MME type markers. And similar to what we showed with the human adipose tissue macs, we could isolate mammary adipose tissue macrophages from obese mice and take their medias and treat murine triple-negative breast cancer cells, and also show induction of phospho-STAT3 signaling within the cells. In this experiment, we actually were able to get a lot more media than from the humans, so we could also test for what cytokines that are elevated in the MME type adipose tissue macrophages might be driving this. And we used inactivating antibodies and could show that inactivating IL-6 was able to attenuate this signaling cascade, while inactivation of any of the other cytokines that were elevated did not. So it seemed that IL-6 was playing a very important role in driving this. Next, we wanted to see whether obesity was able to promote tumor formation in a triple-negative breast cancer syngeneic model. So in this experiment, what we did is we took female C57 black 6 mice. We fed them either a low-fat diet or high-fat diet to establish pre-existing obesity. And that can be shown here, that following 12 weeks, but before cancer cell injection, you see an increase in body weight, fasting glucose, and expansion of adipose tissue depots in the mice. At this point, the mice were injected with varying numbers of TNBC cells, and then we continued low-fat and high-fat feeding, and then monitored tumor incidence over a 9-week period. And what we observed with two different types of TNBC cancer cell lines was that if the mice were obese, you needed much fewer cells in order to form a tumor. And this sort of is referred to as a limited dilution assay, which is an in vivo functional test of cancer stemness. We also performed this exact same experiment, but only after 4 days of high-fat feeding, because we wanted to get the mice to a state of nutrient excess, but not have the pre-existing obesity. And so after 4 days, not surprisingly, the mice had similar body weight, similar glucose, and adipose tissue had not yet expanded. And then when we injected cells into those mice, we did not observe that same induction of tumor incidence. And so this idea then is saying that it's not just nutrient excess, but it's changes in adipose tissue biology that are driving this. And the macrophages might be a very key component to that. And so to test that idea, we also then compared wild type to the myeloid cell NADPH oxidase 2 knockout, and could show that the NOX2 knockout was able to attenuate that obesity-induced tumor formation, again, with both cell types. In addition to doing that, the NOX2 knockout was also able to attenuate inflammation, as well as MME markers in the adipose tissue macrophages, and was also able to attenuate the ability of those macrophages to induce phospho-STAT3 signaling in the cancer cells ex vivo. Now, of course, we had run all sorts of other functional tests like the tumor sphere assay, but I'm not going to show you all of that data today. And so this kind of led to the following model that, just like is observed in visceral and other subcutaneous depots, these macrophages do accumulate in obese mammary fat, and through free fatty acid exposure in that fat are able to drive IL-6 expression through NADPH oxidase 2, and then that IL-6 signals through GP130 to induce STAT3 phosphorylation and increase TNBC stemness and tumor formation. And so, these two studies really highlight the importance of chronic inflammation in insulin resistance, as well as in comorbidities in patients with obesity, such as breast cancer. And so moving forward, where we're headed with all of this is we have emerging data that suggests that in addition to all of these other mechanistic differences that you see down here at the bottom, we're now actually able to show that the mechanism by which NF-kappa B is getting activated by free fatty acids is not the same as the mechanism by which LPS and other bacterial products is activating NF-kappa B. And we've started to delineate this pathway, identified several components of it, and could show at various stages that we can block inflammation from macrophages exposed to fat without blocking inflammation of macrophages exposed to bacteria. And where we're headed with this is to start developing new anti-inflammatory approaches that can block all of these cytokines from metabolic drivers while leaving them intact from bacterial drivers to preserve host defense. And we're hopeful that we can get to a point where we can get to a therapy that can effectively eliminate chronic inflammation without predisposing to opportunistic infections. However, that data is still emerging, and it's just a little too early to share. We want to make sure we repeat everything and get everything right before disclosing everything. And so with that, I'd just like to thank all of the people that worked on this, as well as the funding. I'm going to highlight several people here. Brittany Coates was a graduate student. She was the first author on the cell metabolism paper and cell reports paper. Payal was a graduate student that was mentored by myself and Marcia Rossner, and she was the first author on that triple negative breast cancer paper. Alex is also a former graduate student, and her work was on looking at the relationship of the MME-like macrophages with insulin resistance in patients. And then, of course, all of the collaborators that I've mentioned throughout. I guess I forgot to mention that Pradeep Singh was the collaborator that we worked on for cystic fibrosis. And Peter Billing was the bariatric surgeon that provided us access to the human visceral and subcutaneous adipose. And all of this work was funded by an R01 from NIDDK. So with that, I'd like to end and take any questions.
Thanks, Lev. That was very fascinating. I can see a lot of tremendous upside and potential to that. If anyone has a question, would you please use the reactions to raise your hand to start, and then or type it in the chat, and we'll go from there. While people have a chance to do that, I have a few questions, so I'll ask one of them. Did you? So to begin with, this NOX2 GP91, can this be targeted therapeutically?
I don't think so. So TLR2 and NOX2 were our first sort of proof of concept of just the specificity. But NADPH oxidase 2 generates reactive oxygen species, and while it doesn't support inflammation in bacterially challenged macrophage, it does support killing by a bacterially challenged macrophage. And so patients with NADPH oxidase 2 mutations, they suffer from chronic infection. So this would not be a good target. And also TLR2, in a similar way, while it may not be important for LPS, it is an important recognizer of other types of pathogens.
Okay. Oh, Ron, go ahead.
Okay, great. That's a really exciting talk. I enjoyed it very much. I wonder if you could comment briefly on this so-called obesity paradox, whereby obesity seems to be associated with better outcome for some cancer types, and melanoma probably is the one that come to my mind, and that also seem to those patients seem to do a better job in standard anti-cancer immunotherapy.
Yes. I think, and obviously I don't have any direct data, so I'm just sharing my thoughts on this. I think when I think about obesity, I think about systemic changes as well as local changes, right? And so one of the things we've learned in looking at this MME phenotype, for example, is that we see it in adipose tissue, but we don't see it in liver or other tissues that don't have fat. And so I think when it comes down to the different cancer types, I like to think about what impact obesity has on that local tissue. And some of those inflammatory changes could be tumor promoting. Other inflammatory changes could be tumor opposing or checkpoint inhibitor enabling. And so I think we need to think about this more specifically in terms of what organ are we talking about? What are the changes in that organ that are being driven by obesity? And how does that interplay mechanistically with the specific cancer that we're thinking about from these epidemiological type associations?
Thanks for your insight.
Yeah.
All right, there's a question in the chat from John Rogers. Is there any work going on with SPM, so specialized pro-resolving macrophages, for help in resolving inflammation?
Yeah, we haven't been doing that. That's obviously an exciting area as well. One can inhibit the inflammatory component or raise the anti-inflammatory component, but that's not an area that we've been focused on.
Okay. I'm curious, in some of the earlier time periods, after the high-fat dieting, before the full 16 weeks,
yep.
Did you see if adipogenesis was altered at all? So one other thing I'm thinking about is the health of the adipocytes as they get bigger. And I didn't know if in your knockout models, if it appeared that maybe the adipose tissue was becoming, if the adipocytes in general maybe were dying sooner, if a lack of adipogenesis was leading to accelerated dysfunction of the adipose tissues.
Right. We didn't look at it directly, but we did kind of quantify cell number and cell size in vivo. And what we observed, Brett, was that 8 weeks was just insufficient to induce enough hypoxia to drive adipocyte death. And so you can think of adipose tissue as a tumor too. As it grows, the vasculature accommodates for that growth. But once it gets to a certain size, the vasculature can't keep up with the tissue growth. And now you start seeing hypoxia. So I didn't show the data, but if we looked at 8 weeks in wild type mice, okay, and compared it to 16 weeks in the wild type mice, at 8 weeks, we saw almost no crown-like structures. We saw no induction of hypoxia and no induction of caspase as 3 indicative of apoptosis. At 16 weeks, now we started seeing some crown-like structures, increases in caspase 3, but that was tremendously magnified in the knockout. And so the way I'm interpreting it is that the defect that occurs with the NADPH oxidase 2 knockout is not needed in terms of lysosomal exocytosis, is not needed at 8 weeks because the cells aren't needed to be cleared at that point. But at 16 weeks, now the cells need to get cleared because there is significant death, and now their inability to clear them is exposing a defect that was just not present earlier on.
Okay, great.
Yeah.
Paul, do you have a question?
Oh, yeah. Sorry. Yeah, thanks, Brett, and thanks, Lev, for that talk. Yeah, and I just want to say I really appreciate the your efforts and the great publications and sort of the just general shifting in paradigm in terms of how the research is, being approached with regards to understanding inflammation in a disease-specific manner. So I really appreciated that, and from a rodent to human perspective, I think it's really important work. So thank you. And I think the one thing that I'm thinking about in the context of chronic inflammation or systemic inflammation, in terms of how I think of it, obviously, I'm not an immunologist, but I think just in general, I wanted to get your insight with regards to, especially from these transitions that likely occur with obesity and diabetes, pre-diabetic or pre-obese, with how you view what's happening to macrophages and with regards to adaptive immunity, and how T cells and T cell memory may be contributing to how those macrophages are behaving or how there are changes in the phenotype with us. So in terms of how do you view that component, which in the larger context of your studies and when you're putting these together? So it's, I mean, you sort of think with regards to how T cells behave and sort of different antigen and immunological memory, one would, and I'm guessing there are people that are looking at this side of the thing, but in terms of, it seems like in terms of therapeutically, we would need to bridge that gap to understand whether or not insulin resistance is causing a antigen specific phenotype that could be affecting innate or acute or chronic macrophage phenotypes. Yeah, so it's sort of, I just wanted to get your take on that in terms of how you view that component.
I think you make an excellent point, and one of the I think exciting aspects of working on innate immunity more generally is that it's kind of the bridge between the tissue dysfunction and the adaptive system as well. So not only can macrophages and dendritic cells impact the tissue cells directly, they can also impact the adaptive system, which then can interface with it as well. We haven't looked specifically in obesity, but we've done a lot of work on this in cancer, where we've been leveraging the innate immune system to get to the adaptive immune system in different and unique ways. And so I definitely think that your thoughts are on point. In terms of specific thoughts in adipose tissue, it's hard for me to comment because we haven't been studying that aspect in obesity. But in cancer, we're starting to find, we just published a paper showing that one way that macrophages can influence the adaptive immune system is through the over-degradation of tumor antigens. And so T cells, before they can get exhausted, they need to get trained. And macrophages seem to be lowering the ability of the innate system to activate T cells in tumors. We haven't looked at this directly in obesity, but I think sort of thematically, those types of studies are becoming important when you start thinking about how these cells are contributing to systemic and tissue dysfunction on a more global level. But I agree, like the lipid antigen work is very challenging. And I think there's been some antigens identified that can, signal, I think through CD1C, if I'm not mistaken, to activate, but we have not been engaged in that work.
Okay, yeah, thank you very much. Great talk.
All right, Lev, I have another question for you. I'm just thinking of the flip side of some of your work. You really nicely showed that more of a classically activated macrophage has its own surface marker profile, and the MMEs have a different one. And I know you focus a lot on the MMEs, but I'm wondering if it's possible to use that other LPS surface profile to assess maybe the cleanliness of inflammation. I'm almost thinking, envisioning assessing the severity of infection versus whether or not it looks, an injury or something is starting to clear up, or, systemic inflammation.
No, that's exactly one things we can start thinking about, is using them as biomarkers to track flares of, for example, in cystic fibrosis patients, their disease is not stable. They go through periods of flares and then better times. And so one can perhaps start thinking about using them as biomarkers to predict those types of events. But I think I didn't show this data, but taking a step back, we also in that 2014 paper showed that bacterial products that signal through TLR2 also induce inflammation very highly, but don't express markers of M1 macrophages. And so I think, kind of a one take-home message from me from that work was that the way we define inflammation is through cytokines. The cell surface proteome isn't telling you whether you're inflamed. It's probably giving you insights into how you got inflamed. And my feeling is that a gram-positive bacteria, a gram-negative bacteria, fat, atherogenic lipoproteins, all of these things are known to induce cytokines, but they're all likely to be reported by completely different cell surface markers. And I think this kind of concept is created some confusion in the field, because we've taken markers identified through bacterial exposure and then tried to go look for them in vivo, oftentimes with no successful outcome when you're looking in sterile inflammatory disease. And so I think there's more to it, and the specificity doesn't end between just fatty acids and bacterial triggers. I think different bacterial triggers also have that same dichotomy in terms of the markers that will report it.
Great, thank you. Well, I don't see any other questions, so thank you very much for your time. This was an excellent talk. The subject was very fascinating. We definitely appreciate you sharing this with us. And for everyone here at GW, we look forward to seeing you at next month's cross-disciplinary seminar series. Thank you very much, Lev. Really appreciate it.
Thank you so much. Yeah.